Hello, my name is Angel Craig, and I'd like to present to you a project on how genetic variation in cottonwood trees influences the structure and diversity of dependent arthropod communities on different cottonwood hosts. Our project was led by Carl Jarvis, in collaboration with myself and Rebecca Berzik-Parents, and uses phylogenetic analyses to bring insight into community structure and diversity. In our project, we wanted to build on work at Northern Arizona University in the field of community genetics. Findings of the Community Genetics Group at NAU showed that genetic variation in foundation cottonwood trees can have important community and ecosystem consequences. Different cottonwood genotypes have been studied in an ecosystem where Populus fremontii and Populus angustifolia hybridize to produce progeny that span a broad range of traits. We wanted to study these communities through the lens of phylogenetics, the historical evolutionary relationship among species. By studying the phylogenetic relationships of arthropod communities on both pure cottonwood species and their hybrids, we can gain deeper insight into the mechanisms that influence community assembly. Our overarching hypothesis is that hybridization in cottonwoods drives differences in the phylogenetic diversity and structure of arthropod communities. Our approach to studying biodiversity requires different types of data. We made use of an existing large data set of arthropods that used community ecological metrics. We analyzed the data to evaluate arthropod communities within a phylogenetic context. We found major differences in phylogenetic patterns of arthropod communities on hybrid versus parental hosts. Hybrid cottonwood arthropod communities are more distantly related to one another and have greater phylogenetic diversity. In contrast, the communities of arthropods found on the parental hosts had the opposite pattern. They were much more closely related. Our results suggest support for our original hypothesis that hybridization in cottonwoods drives differences in phylogenetic diversity and structure of their dependent arthropod communities. Our findings of unique phylogenetic patterns for arthropod communities on hybrid cottonwood suggest that hybrids may play a unique role in the formation of ecological communities, a role that likely extends to the entire ecosystem. Additionally, understanding how plant genes may influence community structure of organisms can bring important insights into what impacts genetically modified crops may have on the environment. Greater awareness of how genetic interactions influence the makeup of the natural world can help us better understand evolution, community dynamics, and how we can best preserve and maintain biodiversity globally.